First, let's know what is the difference between mucus and mucin. Mucus is a substance that is secreted by various snail species by their individual mucous pedal glands, and it covers the entire animal's exterior that will play a vital role in the survival across diverse environments. While mucin is a specialized protein with highly glycosylated proteins that have the ability to stick to the surface and retain moisture and resist microbial attack. It also contains bioactive substances that are used in medicine. For example, cornoaspersum is one of the snails that produce viscous secretion called mucin that contain multi-purpose bioactive substances that are used in dermatology and medicine that support antibacterial and anti-inflammatory. There is a wide variety of bioactive substances that can be found in snail mucin. One of them is hyaluronic acid and allantoin. These are the 2D and 3D structure of hyaluronic acid. Hyaluronic acid is known as humectant with high capacity to retain water and serve to moisture to the skin. This is the 2D and 3D structure for allantoin. Allantoin is a natural substance that promotes cell growth and are used in cosmetic and wound care products. It also plays a major role in enhancing fibroblast activity and reducing oxidative stress. The mucin have a natural milky adhesive polymer owing to its biocompatibility. It is a clear and pale yellow in color. The density for the mucin is 102 gram per milliliter and the pH is roughly 4.80. It is biodegradability in the living system and it is a non-antigenic and a non-toxic.
Now, let's see how snail mucin is synthesized. The process begins by carefully cracking the snail shell and gently remove the fleshy body with a sterile metal rod. Any waste or excretory material is carefully cleaned away. Next, 10 grams of snail tissues are washed repeatedly in 250 ml of water. The slime is squeezed out and collected. This washing step is repeated 3 times, which will result in 1 liter of mucus-rich solution. To isolate the mucin, we add 2 liters of acetone to the pooled washings. This causes the mucin to precipitate out of the solution as a solid. The precipitate is filtered and then lyophilized or freeze-dried, which will produce a brownish flakes. These flakes are then blended into a fine powder. The mucin powder is stored in an airtight container to preserve its quality. For comparison, the entire process is repeated using 2% alum solution as an alternative precipitating agent. Next, we move on to the application of snail mucin. First, snail mucin can act as oral drug delivery. Snail mucin can increase the solubility and bioavailability of poorly soluble medications when administered orally. Its mucoadhesive qualities will guarantee extended retention in the gastrointestinal system, improving the effectiveness and absorption of medications. Mucin's sticky properties enable improved localization and regulated medication release in the stomach during the development of gastro-retentive drug delivery system. Next, snail mucin can act as an anti-tumor agent. Snail mucin has shown therapeutic potential against melanoma, which is one of the dangerous skin cancers. As treatment resistance is common for this cancer, there is an urgent need to find effective novel approaches for treating melanoma. A study of H. apersa, mucus on melanoma cell lines, reported that snail mucus decreased the viability and inhibited the metastasis of melanoma cell. Additionally, the inhibition of metastasis was achieved by blocking integrin function and expression and thus inhibiting the cancer from being able to grow.
Snail mucin contains a various bioactive compounds such as glycoproteins, allantoin, and hyaluronic acid which contribute to skin health. It helps to improve skin hydration and elasticity, while it is an anti-inflammatory and antimicrobial properties that can reduce acne and skin irritation. Snail mucin is also known as a promoting wound healing and tissue regeneration by stimulating collagen production. Overall, Snail Mucin is considered as a safe and widely used in skincare product with a minimal adverse effect. Current research on snail mucin goes beyond cosmetic application and explores its potential in medical and biomedical field. According to studies reported by Headline and Medical News today, snail mucin has shown a promising effect in wound healing by accelerating tissue repair and improving collagen deposition. In addition, findings published in the International Journal of Biological Macromolecules and MDPI indicates that bioactive peptide in snail mucin may support tissue regeneration and exhibit antimicrobial activity. However, most of these findings are based on the laboratory and early stage study. Therefore, further large-scale clinical trials are still required to fully validate its medical application.